We are continuing on with the Chapter 5 lecture notes. We left off at slide 18. We were looking at cell signaling or cell communication. So I'm just going to review. So what we're looking at here is that this is the outside of the cell, this little part in the corner, in the top left corner. And then we have the plasma membrane of the cell. You can see the phospholipid bilayer. And then you can see the orange protein molecules within the phospholipid bilayer. And this particular protein is a receptor protein. So what we're seeing is a signaling molecule, like a hormone, is binding to the receptor protein. And we say that in that way that the cell has recognized it whenever it binds to a receptor protein. And then what you're looking at here with the square and the triangle and the circle, this is just representing a series of chemical reactions that occur. One leads to the next, leads to the next. It's called a transduction pathway. And at the end of that transduction pathway, a protein is activated. The protein may be a structural protein, and it may alter the shape or the movement of the cell. The protein that is activated may be an enzyme, and that enzyme may catalyze a chemical reaction. It may be a gene regulatory protein, meaning that it will turn on or turn off the expression of a gene, that kind of thing. So this is just an overview of one way that cells communicate and that cells respond to signals. Okay, so, all right. Now we're going to move on and talk about the transport across the plasma membrane. So we already know the plasma membrane is selectively permeable. That means it allows some molecules to cross and some it does not permit to cross. Molecules that can freely cross the plasma membrane through the phospholipid bilayer are going to be lipid soluble. In other words, they are not soluble in water. So these molecules are going to be small, they're going to be non-charged, and if they're covalent, they'll be non-polar. The molecules will not be soluble in water, so they won't have a charge and they won't be polar because water is polar. So they're non-polar, non-charged, and they are small. The two gases that we breathe in and out, carbon dioxide and oxygen, diffuse across the phospholipid bilayer this way. Glycerol is another one, alcohol is another one. And the molecules move with their concentration gradient. So they're going to move from where their concentration is high to where it is low. If the concentration inside the cell is low, and the concentration outside the cell is high, then these molecules are going to move into the cell through that phospholipid bilayer. Okay? Now, water is going to move through the plasma membrane through these specialized membrane proteins called aquaporins. And here's just a blown up image of the phospholipid bilayer with some of the proteins. This protein that's shown in green has a channel through it, so this is a channel protein. And molecules that will not be able to move through that phospholipid bilayer are going to be molecules with a charge. Charged molecules, ions, which are atoms that have a charge, like sodium ions and chloride ions, potassium ions, and polar molecules will not be able to move through that phospholipid bilayer. But water can move through aquaporins, and sometimes a small amount can even move through the phospholipid bilayer just because water is so small. It's technically polar, and so it shouldn't be able to, but because it's so small, some water molecules will get through that phospholipid bilayer. But mostly water moves through the aquaporins. And then we have non-charged molecules, whatever this purple molecule is right here, this little chain. It's non-charged and non-polar, so it can move freely through the phospholipid bilayer. Macromolecules are too large. Macromolecules are just large molecules. This green molecule here looks like a large protein, and it's just too large to move through the phospholipid bilayer. This is a table, table 5.1, that lists the name of types of transport. So diffusion is a type of transport where molecules move toward their lower concentration. Or another way to say it is they move from their high to their low concentration. The requirement is that there is a concentration gradient or difference. Concentration gradient just means a difference in concentration from from outside to inside the cell. And examples of molecules that pass through the cell by diffusion are lipid-soluble molecules and gases. Now, facilitated transport is also a type of diffusion, but the molecules that are moving this way are either polar molecules or they have a charge, and that means they're soluble in water. So they have to move through a channel or a carrier protein. They're also going to move toward their lower concentration, just like in simple diffusion, but they're going to have to move through a channel or a carrier membrane protein. Examples would be some sugars and amino acids. Then we have active transport, which requires ATP energy and also a carrier protein. And molecules, instead of moving with their concentration gradient from high to low concentration, they're going to actually move from their low concentration toward their higher concentration. And sugars and amino acids can do this depending on their concentration gradient. Ions also can move by active transport. All right, and then there's bulk transport, which is going to be called endocytosis or exocytosis, and this utilizes a vesicle that fuses with the plasma membrane. So this is going to be for substances that are too large to move through the phospholipid bilayer or through a carrier or channel protein. All right, here are the notes on passive transport. Passive transport means that molecules are going to move from their high concentration to their low, and energy is not involved. The passive transport does not require energy. So our first example of passive transport is simple diffusion. I'm going to skip this because we're just going to concentrate on diffusion across the plasma membrane, not diffusion of a dye in water. So I'm going to skip that part. But what you're looking at here is an image of this part is the membrane that surrounds the air sacs in your lungs called alveoli. And then this is a capillary, a blood capillary, which has very, very thin membranes. So when oxygen is breathed in, when you breathe in oxygen gas, that oxygen gas ends up in the air sacs of your lungs, in the alveoli of your lungs. So the concentration of oxygen is very high in those alveoli. It's low in your blood. So oxygen is simply going to diffuse across the phospholipid bilayer of the plasma membranes of the alveoli and the capillary. And it's going to diffuse from the lungs into the blood. And then if we were to see carbon dioxide up here, it would be opposite. Carbon dioxide builds up in your tissues. So the concentration of carbon dioxide would be high in the blood and low in the air sacs of the lungs. So carbon dioxide would actually move in the opposite direction from the capillary into the alveoli. But they're both moving by simple diffusion through the phospholipid bilayer. Now, a special case of diffusion is osmosis, and that is when water diffuses across the plasma membrane. And sometimes this happens because other molecules that are dissolved in water aren't able to move across the membrane, but water is more freely able to move. So if you have a solution, for example, that's 10% salt, and we're using a thistle tube here. This is called a thistle tube. Thistle tube in a beaker of water, and then this red membrane represents the plasma membrane. It's permeable to water, so it allows water to move across, but it does not allow the salt that's dissolved in the water to move across. So if you have a 10% salt solution in the tube and a 5% salt solution outside the tube, there's a higher concentration of water in the beaker. So the water will move from where water's concentration is high to where water's concentration is low. So that's what you're going to see happen, and then you see the water level rise in the thistle tube because water was high in the beaker and lower in the tube. So water moves from its high to low concentration. But don't let the numbers confuse you. The numbers are the percents of solute in the water. And another way that you can remember what happens and where water moves, whether it moves into the cell or out of the cell, is that water follows, hopefully I can write this, water follows salt or whatever solute, water follows solute. Gosh. Okay, hopefully you can read that. Water follows solute. Water moves to the higher solute concentration. If you remember that, you'll always know which way water moves. Water is going to move toward the higher solute concentration. So that is the reason that water moves into the thistle tube, because that's where more solute is dissolved. Okay? Now, what happens in animal cells when an animal cell is placed into a solution that is isotonic to the inside of the cell? That means that the solution has the same solute concentration as inside the cell. In that case, the cell is not going to change size. It's not going to shrink. It's not going to swell, because the water that comes in is equal to the water that goes out. So it overall will not change size. So an isotonic solution is a solution that has an equal concentration of solute as what's inside the cell, as the cytoplasm. In a hypotonic solution, that means the solution has a lower solute concentration than inside the cell. So for example, in this beaker, the beaker had a 5% concentration of solute, which was lower than the concentration in the thistle tube, which represents a cell. So the solution here would be hypotonic. The solution in the beaker would be hypotonic. And what happened? The thistle tube, which represents a cell, took on water. Water went into the thistle tube. Same thing that happens in animal cells. The water will move into the cell, and it could even swell and burst, and we would call that lysis. If the solution is hypertonic compared to the inside of the cell, that means it has a higher solute concentration. It's much more salty or much more concentrated with solute than what's inside the cell. Water's going to follow that solute. Water's going to move out. So when it does, the cell will shrink, and that is called crenation. In plant cells, you see the same basic thing happen, but there is a cell wall that stops that cell from bursting. So in an isotonic solution, you won't see much change in the size of the plant cell. When the plant cell is placed in a hypotonic solution, water will move in, the cell will swell, but it will push against the cell wall. It won't burst, and this is called turgor pressure. And all of those organelles, especially there's so many chloroplasts, they're going to get pushed up to the edge, right up against the edge of the cell wall. And that's something you would notice under the microscope. Now, if the plant cell is placed in a hypertonic solution, then the plasma membrane will pull away from the cell wall. So the cell itself will shrink, but the cell wall is a rigid structure and it won't change. So you would see the plasma membrane pull away from the cell wall, and you'd see all the organelles kind of move toward the center here, because now the cell is smaller. So all the organelles are going to be bunched up together. But the plasma membrane, this blue structure, pulls away from the cell wall. See how it's up against the cell wall here? But here this is the plasma membrane and this is the cell wall.